Hello students, welcome to EPG Pathshala. I am Mrs. Bhojwani. Today we are going to discuss about a module on in vitro production of haploids under the paper Plant Biotechnology and Crop Improvement. In this paper, we'll try to learn what are haploids, what is the importance of haploids, what is the frequency of occurrence of haploids in nature, and the in vitro techniques to produce haploids. The haploids plants are plants with a single set of chromosomes, or you can also say gametic number of chromosomes. And these are important in genetics and plant breeding because they have only one set of chromosomes. And therefore, all the genes are expressed morphologically seen. And one can detect mutations, or after hybridization, one can detect all the characters because diploids produced from the haploids are homozygous. So today we are going to learn the three important techniques of haploid production in tissue culture. It is important to produce in tissue culture because the natural occurrence of the haploids is extremely poor, and therefore the breeders could not use this in their research. Now we have at least three techniques, in vitro techniques, to produce haploids by Anther and pollen culture, which is known as Androgenesis, by the culture of unfertilized ovules, ovaries, or florets, which is Gynogenesis, and the third one is a distant hybridization followed by embryo culture. Each technique has its own advantages and limitations. Most popular in vitro technique is androgenesis because you have very large number of haploid cells available in an anther. For example, you have 40,000 pollen grain within an anther of tobacco. But it has its own limitation because in cereals, most of the androgenic plants are albinos, which are no good because they can't survive in nature. But on the other hand, if you produce gynogenetically by the culture of unfertilized ovules or ovaries, a very large number of green plants appear which can be used. Therefore, people prefer to produce cereal haploids by gynogenesis. Even better technique for the production of haploids of cereals is distant hybridization followed by embryo culture, in which the plant for which you want to produce the haploids is pollinated by another plant, in which cross the fertilization takes place, but the chromosomes of the second parent are selectively eliminated, and they are left, the egg or the proembryo is left with only the chromosome of that particular plant. However, the problem is that in this case, the embryo aborts after 10 days, and there comes the role of tissue culture. You can excise the immature embryo before abortion and culture and rear full plants. And this is a very important technique for the production of haploids of cereals, and people prefer to do that. And all the plants agree. Production of haploids in tissue culture and crop improvement. The first question is, what are haploids? And as you know, in genetics and plant breeding, it's a very common topic. The haploids are plants with gametic number of chromosomes, or we can also say one set of chromosomes. And the question is, why we need haploids? Haploids are extremely important for plant breeding and genetics because the breeders wants to achieve homozygosity, and the haploids permit to achieve homozygosity in one step, which takes several years under the ordinary conditions. Unfortunately, in nature, there are no reliable and reproducible methods to produce haploids. As I said, the natural occurrence of haploids is extremely low. For example, it's said that about 0.001 to 0.01%. And there was a report in 1922 where Blakesley et al. Got the haploids by pollinating the tobacco plants, datura plants, with irradiated pollen grain, and he got some haploids. But nobody could repeat this technique. Therefore, there is no reliable or dependable, reproducible in vivo method to produce haploids. Therefore, the importance of in vitro production of haploids. And in vitro, the haploids can be produced from the haploid cells. The question is, where are the haploid cells available in the plant body? Because most of the cells of the plant body are diploid. The only places where haploid cells in the plant body are available are the gametophytes, because the diploid cells in those tissues undergo meiosis and produce haploid cells. And these haploid cells are both on the male side and female side. So we have male gametophyte, the pollen grain, and the female gametophyte, the embryo sac, where the haploid cells are available. And if these cells can be triggered under artificial conditions of in vitro techniques, one can induce those cells to form haploid plants. Therefore, in 1964, when the two scientists at the Delhi University were trying to study the physiology of meiosis in the cultures of anthers, they discovered that after some time, the anthers dried up from the outside and burst open, and from the inside, the green plants emerged. This was in 1964, and this was a breakthrough. In 1966, they confirmed cytologically that these plants which arose from inside the anther were haploids. And this discovery changed the entire thinking of the geneticists and plant breeding, and they jumped over in this area because they were waiting to find a reliable method to produce haploids which can be used in plant breeding and genetics. But until then, they didn't have, and therefore these haploids were not fully exploited. Now, the question is, where are the haploid cells in plants? You know that the plant body cells are diploid, which means there are two sets of chromosomes. The only cells in the plant body that they have haploid number of chromosomes or one set of chromosomes are the gametophytic cells. On the male side, they are pollen grains. On the female side, it is the female gametophyte, which is called the embryo sac. They contain haploid cells. So one can tap those haploid cells and regenerate plant. You will probably study in tissue culture in this course about the phenomenon of cellular totipotency, which was, until 1964, was restricted to somatic diploid cells. Now, it is being extended, and we now can say that the haploid cells, or the gametic cells, are also totipotent, which means that they can also regenerate full plants. And therefore, here is an opportunity to produce haploids at will in large numbers. There are three in vitro methods to produce haploids: androgenesis, gynogenesis, and distant hybridization followed by embryo culture. Of these, androgenesis is most popular because an anther, say about tobacco, each anther has about 40,000 pollen grains, and all are haploids, which means that you have 40,000 potential grains or cells inside an anther to form plants which will be haploid. Whereas on the gynogenesis side, there are at the most 8 cells in a female gametophyte. So the number, the frequency of cells which can form the embryos is extremely low. Therefore, androgenesis has been the most popular method to produce haploids in tissue culture or in vitro. Now, there are two methods in androgenesis to produce haploids. Initially, as in the case of Datura in 1964, when Guha and Maheshwari were working, they cultured anthers. And you culture the anther on a standard tissue culture medium, and the pollen grain inside will respond to it. Soon people realized that once you have induced the pollen grain to form plants, you don't know what is happening inside the anther culture unless and until such time that the anther bursts open. And also, the treatments that you apply to anther are first come in contact with the anther wall, and then they enter the anther wall and reach the pollen grains. And one does not know how these chemicals are modified by the time it reaches the pollen grain. Therefore, people thought that it might be worthwhile trying to see if the pollen grain can be isolated from the anther grain and cultured in isolation. And that's what is known as microspore culture. And in late 1960s, as French scientists in the laboratory of Professor Nitsch were successful in culturing isolated pollen grains of tobacco and raising full plants from it. The technique of isolated microspore cultures involves, it's a very simple technique, that you take the flower buds, isolate the anthers, put them in a liquid medium, and crush them with a pestle. And once this is crushed, the pollen grain will come out and float into the medium, which is then filtered, and the filtrate, which is carrying the microspores, and the filter paper carries the rubbish on the top. The filtrate is then centrifuged in a tube, and the all fine rubbish and the microspores settle down, which are then purified by putting on gradient centrifugation, and the purified portion is collected, suspended in a liquid medium, and plated. So these microspores are generally cultured in liquid medium. Now, what are the factors that affect androgenesis? The most important factor is the genotype, the genetic makeup of the donor plant, because sometimes the variation is so large that some varieties will show no response and the other varieties are most responsive. For example, in the case of brassicas, the Brassica napus is highly responding for androgenesis, whereas Brassica gensea, which is the Indian mustard, shows poor response. As compared to this, one can get the haploids, but the responses compared to the napus is lower. Sometimes the differences also occur intra-varietally, because variety is also made up of heterogeneous plants. The other factor to take into consideration is the physiological status of the donor plant at the time of excising the microspores. Generally, the pollen grain from the first flush of flowers give better response than those from the older plants. The culture medium, of course, in tissue culture, all responses are dependent on culture medium. The culture media that are used for anthropollen culture are low in salts, and the microspore culture are even lower from the anther culture medium. You will see a composition of an anther culture medium and an N6 medium and a microspore culture medium, which is the NL medium. And you will see that the microspore culture medium is low in salts but richer in organic compounds, apparently because in anther culture, the pollen grain inside the anther get nutrient from the anther wall also, and the organic matter is supplied. But the isolated microspore culture do not have the benefit of these factors from the anther wall, therefore they have to be enriched with organic matter. The stage of pollen at which the anther is cultured or the pollen grains are cultured is very critical. You know that the development of pollen grains, the microspore initially has a nucleus centrally placed in the cell. As it develops, the nucleus is pushed to one side and an asymmetrical cell division takes place. And it cuts a small generative cell and a large vegetative cell. And once the two cells are formed, gradually the gametophyte-specific information is transcribed in the cytoplasm. And once it is advanced to that stage, the pollen grain are irreversibly committed to form gametophyte. Therefore, the best stage in most of the cases is just at or just after the first division of the microspore when the generative cell and vegetative cell are formed. There are a number of treatments that have been used to trigger the sporophytic development from the pollen grains. And these pre-treatments include such as the temperature shocks. In large number of cases, subject cultured anthers or the microspores to 4 degrees Celsius or to 6 degrees Celsius for 2 to 3 days before transferring them to a standard temperature of 25 degrees Celsius is found promotive for androgenesis. But there are on the other side some plants, such as all brassica species. They required a higher temperature shock, and they are subjected to a temperature of 30 to 35 degrees Celsius. Then there are osmotic shocks. Some people are putting mannitol. There are nutrient starvation shocks and centrifugation and similarly colchicine treatment. These are the pre-treatments that promote the induction of androgenesis. And as you will see, a large number of these treatments interfere with the normal development of the pollen grains and then promote them to form sporophyte. Once a pollen grain has been induced, the genes which were specific for the gametophyte development stop their transcription. It is the sporophyte specific genes which start transcribing, and the information is formed in the cytoplasm. And the cell is triggered to form a sporophytic cell by developing a typical cellular wall inside the pollen wall, because you know the pollen wall is made up of sporopollen, and the somatic cell wall is made of cellulose. So once the cellulosic wall encloses the cytoplasm and the nucleus, the cell becomes a sporophyte type of a cell and undergoes a pathway of sporophytic development. An illustration of the temperature shock, if you'll see in brassica. If you keep the culture of brassica microspores or the anthers at normal 18 degrees Celsius, they all develop normal pollen grains. But if you keep them at higher temperature of 32 degrees Celsius, these pollen grains will undergo a gametophytic type to develop, a sporophytic type of development, and form the haploid plants. All the treatments, as I said, will disturb the normal development of gametophyte, and more specifically, the cytoskeleton, which includes the microtubules and the actin filament in the cytoplasm, they are distorted. Because these are the cytoskeleton which under normal conditions and normal development of the pollen grain pushes the nucleus to one side for the asymmetrical division. And if these microtubules are disturbed, the nucleus does not move to the center and divides on its own like a typical sporophytic cell. And you'll see this, the division. And once these induced cells are ready to form gametophyte, they undergo cell divisions. Now, the early segmentation of the pollen grains is different in different plants. Generally, the pollen grain divides by asymmetric division and forms generative cell and the vegetative cell. And it is the vegetative cell which gives rise to a haploid plant. This is the most common pathway. The media generally used for anther culture or pollen culture are similar but not identical to those used for somatic tissue cultures. These are generally low salt media, and particularly the ammonium salt. In the case of isolated microspore culture, it is even more important to be careful that the ammonium salt is extremely low because they are directly exposed to the medium and are very sensitive to ammonium salt. However, as compared to the anther culture medium, the microspore culture medium is rich in organic matters, organic constituents, because the factors that in anther culture pollen gets from the anther wall is not available to the isolated pollen culture. And in the medium, generally we use 3% sucrose or 2% sucrose. Here also, a large number of cases we use 3 to 6%, but there are cases which requires a very high concentration of sucrose, such as brassicas. All the brassicas species, the anther culture requires 10 to 13% sucrose. The conditions, the culture conditions that induce androgenesis or gynogenesis are responsible to disturb the normal development of the gametophyte. For example, in the case of pollen development, the cytoskeleton is extremely important for the asynchronous division of the microspore, by which a small generative cell and a large vegetative cell are formed. But when you give the treatments, such a pre-treatment such as cold treatment, high temperature shock, or colchicine treatment, or centrifugation, these disturb the disposition of these cytoskeleton. And this division of this cell, or the microspore, is not a typical asynchronous, asymmetrical, but it could be symmetrical, or the expression of the genes specific to gametophyte is suppressed and the expression of sporophyte specific genes is promoted, and the cytoplasm becomes rich in sporophyte specific information. And this such, you arrest the development of gametophyte and promote the development of sporophyte. In other cases, such as brassica, the pollen grain microspore divides by an equal division, and both the cells participate in the formation of the plant. In the case of datura, both the generative and vegetative cell participate. The only plant where the generative cell gives rise to the plant is Hyoscyamus niger. That's the isolated example. Now, once the induction has taken place, the initial divisions has taken place, and a multicellular pollen grain has been formed. The pollen wall can retain the cells only up to a certain limit, and once it grows beyond size, the pollen grain bursts open and releases that tissue mass. Now, depending on the species, this tissue mass can directly form an embryo, as in the case of tobacco, mustard, Hyoscyamus, or it can develop into a callus tissue, which will then later differentiate plants, as it happens in the case of datura and all the cereals. The embryos that are formed from pollen grain are typical of that plant. Even the suspensor cells can be seen in these embryos. The major problems with these plants in the case of androgenesis is that androgenesis, although it's applicable to a large number of plants, is not applicable to all crop plants. For example, it's not applicable to onion, sunflower, sugar beet, mulberry, cucurbits, legumes. They don't respond. They don't show androgenesis. Secondly, in the case of cereal, there's a very serious problem that the pollen plants show very high frequency of albinos. And you know the albinos don't have chlorophyll and therefore don't survive in the field. So these plants are useless. And the third problem is that sometimes non-haploids also appear in the population of pollen plants that they are formed. Now, once the haploids are formed, the haploids are not fertile. You can imagine why? Because they are not fertile because they cannot have normal meiosis. Because meiosis requires homologous chromosomes. And the haploids has only one set of chromosomes and lacks the homologous. Let us now summarize this module. In this module, we have learned what are haploids, why should we produce haploids, and why specifically in tissue cultures. And I have explained to you that necessity of in vitro production of haploid becomes important because the haploids are extremely important in plant breeding research because they give a quick method of producing homozygous plants. Unfortunately, in nature, we don't have a reliable method to produce them. Therefore, it is extremely important that we are able to produce haploids in tissue culture. And it all started in 1964 with the discovery of androgenesis in Datura. And thereafter, since it was such an important aspect, yet it was limited by non-availability, large number of scientists become interested in this area. And there are a number of books now available on haploid production. There were a number of conferences that held after that. And today, we have over 250 species, plant species, in which haploids have been produced in tissue culture. I've also told you that in cultures, the most popular method to produce haploid is androgenesis by Anther or microspore culture. But because of its certain limitations, there's an alternative method people have used is gynogenesis, in which the haploids are produced by triggering a cell of the female gametophyte. And there is yet another method to produce haploids of cereals, which overcome the problem of albinism and non-haploid, is the distant hybridization followed by embryo culture. The applications, some were expected, some were non-expected. The expected were that we are now able to use these haploids in plant breeding program. A large number of new varieties of croplands have been developed using these plants. And the unexpected was the production of super males of asparagus, which gives you an opportunity to produce 100% progeny with male plants, which was not possible by the conventional method, where you have 50% male and 50% female, and the males have a higher market value, and therefore you get a premium on your product. Thank you very much.